In this video, we will explore the intricacies of cochlear histology, focusing on the structure and function of the cochlea and the organ of Corti. We will discuss topics such as cochlear mechanics, hair cell regeneration, and the role of supporting cells. Join us in unraveling the mysteries of the cochlea. In the video, we will explore the cochlea and the organ of Corti, focusing on cochlear mechanics. The cochlea is a snail-shaped structure in the inner ear divided into the scala vestibuli, scala media, and scala tympani. We will discuss how sound vibrations are transmitted through these chambers, leading to the stimulation of hair cells and the perception of sound. Within the cochlea, there are two types of sensory cells known as inner hair cells, IHCs, and outer hair cells, OHCs. The differentiation of these hair cells is governed by specific molecular mechanisms. IHCs are essential for transmitting auditory information to the brain, while OHCs play a critical role in sound amplification and frequency tuning. The differentiation of IHCs and OHCs involves intricate genetic and epigenetic processes, including the expression of specific genes, interactions with neighboring supporting cells, and the formation of a specialized synaptic connection with spiral ganglion neurons. The cochlear sensory epithelium, or organ of Corti, cannot naturally regenerate after damage, leading to irreversible hearing loss. Research shows potential strategies for hair cell regeneration, including stem cells and gene therapy. Challenges persist, but progress offers hope for future treatments. Hair cell synaptopathy refers to the selective degeneration or dysfunction of the synapses between hair cells and spiral ganglion neurons. Although audiometric evaluations may indicate normal hearing thresholds, individuals with hair cell synaptopathy experience difficulties in understanding speech, particularly in noisy environments. This condition, often associated with noise-induced hearing loss, highlights the importance of synaptic connections between hair cells and spiral ganglion neurons and the maintenance of auditory function. Spiral ganglion neurons, SGNs, are the primary conveyors of auditory information from the cochlea to the central auditory pathways. These neurons exhibit remarkable diversity in their structure and functional properties, allowing for precise encoding and transmission of auditory signals. SGNs can be categorized into different subtypes based on their innervation patterns and neurotransmitter phenotypes. Understanding the organization and function of SGNs is crucial for developing strategies to restore hearing function in individuals with SGN pathology. Otoacoustic emissions, OIs, are sounds generated within the cochlea in response to the stimulation of hair cells. These acoustic signals can be measured using sensitive microphones placed in the ear canal. OIs can provide valuable information about the integrity and functionality of the cochlea, particularly in newborn hearing screenings and monitoring the effects of ototoxic drugs. The analysis of OIs has become an indispensable tool in audiology and has contributed significantly to our understanding of cochlear physiology. Cochlear implants are electronic devices surgically implanted in individuals with severe to profound sensory neural hearing loss. These devices bypass the non-functional or damaged hair cells and directly stimulate the remaining viable auditory nerve fibers, enabling the perception of sound. Cochlear implants consist of an externally worn speech processor and an internal array of electrodes that are strategically placed within the cochlea to maximize speech perception. The success of cochlear implants has revolutionized the field of auditory rehabilitation and has provided a significant advancement in treating individuals with severe hearing loss. Crestin is a protein found exclusively in the outer hair cells of the cochlea. It plays a crucial role in the active amplification of sound within the cochlea, allowing for exquisite sensitivity and frequency selectivity. Crestin undergoes conformational changes in response to changes in voltage, which leads to the electromotility of OHCs. This unique property enables the cochlea to amplify soft sounds and enhance the discrimination of different frequencies. Hair cells possess specialized mechanotransduction channels that convert mechanical vibrations into electrical signals. These transduction channels are located on the stereocilia, which are microvilli-like structures present on the apical surface of hair cells. The opening and closing of these transduction channels in response to mechanical stimuli, such as sound-induced vibrations, lead to changes in the hair cell's membrane potential, initiating the transmission of auditory signals. Supporting cells within the cochlea play crucial roles in maintaining the structural integrity and functionality of the organ of Corti. Disruption of supporting cell function can have profound effects on hair cell survival and auditory function. Various pathological conditions and genetic mutations can lead to supporting cell dysfunction, resulting in hearing loss and impaired cochlear mechanics. Reissner's membrane is a thin, flexible tissue that separates the scala media from the scala vestibuli within the cochlea. It acts as a barrier, allowing for the proper ionic composition and endolymph volume regulation essential for normal cochlear function. Disruptions in Reissner's membrane can lead to conditions such as endolymphatic hydrops, a disorder characterized by fluctuations in ear pressure and hearing loss. Auditory neuropathy spectrum disorder, ANSD, is a hearing disorder characterized by normal outer hair cell function but impaired neural transmission of auditory signals. Individuals with ANSD often exhibit hearing difficulties, particularly in noisy environments, and have abnormal auditory brainstem responses. ANSD can result from various etiological factors, including genetic mutations, neuropathies, or damage to the auditory nerve fibers. Understanding the histological basis of ANSD is crucial for developing targeted therapies and interventions. In summary, the study of cochlear histology helps us understand how we hear. From the mechanics of the cochlea to the processes of hair cell development and regeneration, each aspect is important for maintaining normal hearing. Recent advancements in audiology, like cochlear implants and hair cell regeneration, have been made possible by our understanding of cochlear histology. Ongoing research in these areas offers hope for future progress in treating hearing loss and auditory disorders.